Good afternoon. Thank you very much to the OIS team for giving us a chance to provide an update on ONL Therapeutics. ONL, we've been focused over the last few years on delivering on our commitments to investors from our Series B round, which we closed in the end of 2020. Those commitments specifically were to take our lead compound, ONL 1204, through 3 distinct phase 1B programs. First with retinal detachment, then with geographic atrophy, and then also in open-angle glaucoma. The data over the last few years across those 3 distinct indications continues to look positive, and we're preparing to raise Series C round to be able to accelerate ONL 1204 into phase 2 programs. We've been guided by a strong team. The ONL management team is here, guided by Dr. David Zaks, who's our founder. It was David's discovery in his lab with the relevance of the fast receptor that gave the underpinnings to the company. Also, from our board and investor standpoint, we have several of the investors here today in Focus Capital Partners, Excite Ventures. In addition to several strategic investors, we've had investment from Janssen and Novartis. Both Janssen and Novartis have board observer roles. Their development teams have helped guide our development in a number of ways, in addition to our scientific advisory board. So I feel like we have good support as we move the programs forward. A little background on our technology. ONL 1204 is a peptide delivered IVT injection. And what we see today to be able to block the fast receptor is the intent of ONL 1204. The fast receptor on the surface of a retinal cell, regardless of the disease stress in the eye, whether that's glaucoma, AMD, retinal detachment, the fast receptor is activated, triggers 2 key things: the death signaling of that retinal cell and then the inflammatory signaling. That inflammatory signaling over time releases cytokines and chemokines, which creates a greater recruitment of immune cells to that site of injury, which ultimately brings about greater retinal cell death. ONL 1204, now in humans, in our first human study, demonstrated biological activity on blocking the fast receptor. So we feel in our early clinical programs, we've determined 1204 is doing what we've asked it to do: block the fast receptor and begin to quiet the inflammatory response and the injury in the eye. Importantly, also with regards to FAST, over the last couple of years, we've seen FAST is upstream of all the complement pathways. So we feel it can provide some significant benefits when we look across the board of a number of disease states, specifically in GA. As we look to take ONL 1204 across a number of disease states, we think we provide some really unique positioning. There we go. Unique positioning across each of those disease states. In geographic atrophy, as I mentioned, a mechanism of action that's upstream of some of the positive trends we're seeing with the complement pathways. Also, we're in the clinic today at 90-day dosing. We also see there's potential to stretch that out, but right now, 90-day dosing in the clinic in GA. In glaucoma, we see a neuroprotective treatment that's pressure independent, and we think we can enhance some of the patient compliance challenges in glaucoma with an every 90-day in-office injection to be able to support good physician-patient interactions around glaucoma. And then also in the acute condition of retinal detachment, we see it on top of standard of care surgery to improve visual outcomes. Now, as we proceed into our phase 2 programs, just a couple highlights on some of the early insights we're gathering from our phase 1 data. First, as I mentioned earlier, we've confirmed biological activity on the fast receptor. I feel that's really important given the unique mechanism of action that we can bring towards the clinic. Also, from a safety and tolerability standpoint, we're now in 3 different disease states, 40+ patients, many of those with repeat injections of ONL 1204. The product continues to look safe and well tolerated across those very distinct disease states. And as we look into the data in our numbers today, start to see early efficacy signals above natural history trends in terms of visual outcomes, and also as we see into GA, beginning to see slowing the growth of lesions in this early stage. And so we're excited about the potential to go into our phase 2 programs. I know this is focused on dry AMD, so I just wanted to highlight some of the specifics of our current phase 1B study. This is a design, it's 2 components. We have one that's an open label dose escalation and one that is a 6-month natural history treatment phase. I'll cover the open label dose escalation first. It's a single injection of ONL 1204, monitored over 6 months. Typical dose escalation. We've seen 6 patients through it. We've completed that arm of the study. Important to note, we're starting to see now functional improvement with a single injection of ONL 1204 out to 6 months, and also seeing slowing of growth in lesion in study eye compared to fellow eye. We're excited about that data. Actually, Dr. Charlie Wyckoff is going to present that data in early 2023, so we begin to really pick our heads up and get into the market with some of the data that we're putting forward in AMD. The second component of the study, which is up and running, 30 patients. We're observing patients in a 6-month natural history, and then we randomize and treat with ONL 1204, again, every 90-day dosing in the clinic. We're starting to see similar efficacy trends that we saw in the open label dose escalation. One specific exploratory endpoint we have is with regards to DARC, the technology. You may know the company Novia, the United Kingdom. DARC is detecting apoptotic retinal cells. It's an imaging technology that identifies sick and stressed cells. And what you'll see, this is an image of one of our patients. DARC is identifying those stressed cells on the borders of the lesion where you'd anticipate seeing that growth. And so we're utilizing DARC throughout both the GA study and glaucoma study to be able to identify specifically in GA the progression of the disease, and then also see a potential therapeutic impact much earlier than what you'd see in traditional endpoints. This is a second patient in the study, again, to show the benefits of utilizing a biomarker like DARC to identify those sick and stressed cells on the borders of the lesions. We're excited to see what that's going forward. We're utilizing innovative technologies like that. We're making really good progress in the clinic, and as we look to 2023, we look to be able to accelerate our programs into phase 2, raise a little bit of money to do that. Our team is here. We'd love to take your questions afterward. Thank you very much for your attention.